So how was the weekend, ladies?
That's my friend's birthday party.
Oh, okay. That sounds like fun. Where was the party? At their house or?
Papa.
Oh, at Fapa. Oh, they host parties there? I did not know that. What'd you do? Was it like did you do artsy stuff at the party or is it just a space? Like, did they make it karaoke or anything fun?
We're to dance.
You learned to dance. Okay, so that is a very cool way to do a birthday party, to incorporate all of that fun stuff in there. I like that idea. Was it like a TikTok dance or?
It was to a Freya's High song.
A what?
A Freya's High song.
Oh, okay.
Yeah.
It was fun.
Mhm.
Do you remember it?
Mhm.
So you could do it again when it comes time? Should that ever come? Should the time come for you to do it again? Very good. That sounds like fun. Did you enjoy it, Everly? Yeah, had a good time? It's very cool. I like your hair's fading, but I like the color it's fading too. Do you like it? No, it's good. It's nice. It's fading to kind of a dusky plum color, and I really like it. That's really pretty. Well, that's good. So it was fun. Awesome. Did you give her presents? What'd you give her?
Um, 2 packs of like slushy balls that you can throw at walls or ceilings.
Oh, those are fun. They stick and splat. I love those.
A Roblox gift card.
All right.
A mini verse. Was there another one?
Sticker? You know how to speak. You really do. You don't have to whisper. It's not a secret, is it? Sticker. Okay, cool. So it was fun?
Yeah.
Excellent. That's good. Well, Drew had a gymnastics meet, and Cole left on Sunday to go back to Waco. So off he goes for one more semester, and then he's only got one more after that, and he is done. That'll be so cool to have him be done. But we had the gymnastics meet all of Saturday night. It was late. It didn't start till 5:30, so it didn't end till like 9:45. It was long. But it was okay. It was good. Good to get the first meet out of the way. It doesn't matter how you do at the first meet, you just kind of do. So he didn't feel overly like stressed to do well, just do. I think he has moved my van. Let me make sure that he actually moved it for Ridley. Because he needs to have it out of, yes, he did. Good job. All right. Let's see. Opening up my Ring camera so I can see what's going on outside. Maybe. Doesn't seem to want to open. I don't know why. Oh, that's fun. There were a lot of people at this gym meet though that were falling. I'm telling you, people were falling right and left. I felt like it was a day to watch people fall. Like it was just pretty wild. It's now that Drew's at a pretty high level, the kids at that level start throwing really difficult skills. And it's like, it's awesome when you get it, but it's spectacular when you fail. Spectacular in like a not so great way. So we saw a lot of kids come flying off of high bar and slapping on the floor. And yeah, saw one kid let go of the high bar way too early and fly underneath of it and then land flat on his face on the mat. He actually went, when he got up and shook his head, I was like, this is not pretty, man. But I was telling somebody when we were there that they're little and they're just starting gymnastics. That first year when they fall, everybody, all the parents in the audience goes every time because you're like, oh no, they fell. And then that stops for many years because you just get used to watching them fall until they get to these high levels. And then the falls are like I said, spectacular. So then the comes back and that kid fell off the high bar. The entire audience went, it was crazy. Like, my goodness. Slap right on the mat. All righty. Hey, come on in. How was your weekend? Yeah, good. I was just telling Emerson and Everly that we're gonna have some guests today. We've got some people that are gonna come in and observe class because they're interested in joining us next year. So, do you know if you'll be back next year? Don't know yet? Okay, yeah. They won't because y'all are going to Vickery, right? Yeah, they start middle school next year. So they won't be, but you might. So, all right, well. Right. They're going to public school next year. So that'll be a shift for them. But the rest of us, I think we'll probably be back, hopefully. All right. Well, we're gonna hi, come on in. I'm Danny. Come on in. How are you?
What's
Sit.
up?
Nice to meet you. Hi, I'm Danny. Hi. Sit anywhere you'd like. Come, yeah, you girls can sit up here if you want. Absolutely. And would you ladies mind telling me your name? Addie and
Haley.
Addie and Haley. Okay, this is Emerson and Everly and Evan. This is the class of E's. Emerson. All right, very good. Addie and Haley. At least it's not Emily and Alyssa. We'd had more E's. It would have been more confusing for me. All right, so we are on lesson 19, which means this is the 19th time we've sat down together. And this is our second lesson of our second semester and also the second lesson of our third quarter. We break our school year into quarters and 2 quarters in the first semester, 2 quarters in the second semester. So we are in our third quarter now. And that means we are in our home stretch, right? So this quarter, the unit we're talking about, it goes, basically it starts with the least complex life forms and we move all the way through the most complex life forms. Our first semester, we did a lot of what? Cells, energy, how things function, that kind of thing, ecology, environment, that sort of thing. So now we're into different forms of life. And then our fourth quarter is all like human anatomy and physiology in this class. All right, so we ready to go? Get your binders out, binder out, getting it out. So we have binders, ladies, and they get these at the beginning of the year, and each quarter they get a new packet that leads them through the whole quarter. And it'll have, good job. There's an assignment for every week, and they have a textbook they read along and fill in blanks or answer questions in their binder. And a lot of those questions then show up in class. You ready, gang? All right, so this was your homework assignment, right? Recognize it? Yes? So let's see if we remember from our homework what any of these answers might be. So this is a diagram of a bacteria, and number 1 is pointing right here, and here's your word bank. Do any of you remember what the answer was? You can't see it because your homework was online, but do you remember? Okay, so that was your plasmid. Number 2 is pointing to this big thing right here, this curly thing. Do any of you remember what that was? That was your chromosome. All right, now this one's easy. This is like any other cell. What's all this stuff in the middle, the jelly-like stuff? That's the cytoplasm. Good job. All right, ooh, gotta open up my little cup. So when you answer questions correctly, you get a penny dropped in one of these little cubbies. It's a nice cube tray. And then what do you do with the pennies, gang? Do what? You get Skittles on the way out the door at the little candy machine. Very good. All right. Encourages participation. Does anybody remember what number 4 is? These little finger-like things, those are pili. We're moving through this quick because we're going on to our lesson. This is just homework review. All right, how about number 6? It's all the way in there. So that's going to be, oops, sorry, that went the wrong way. Wrong order. Endospore was 5, 6 is cell wall. All right, let's look at our virus now. Hi, come on in. How are you? If you'd like, there's a seat over here if you want to sit with the gang. And there's a bunch of moms back here. I'm Miss Danny. What was your name? Vander? Okay, cool. I have a Xander over there too. So that's pretty cool. All right, awesome. Well, we are reviewing our homework. They do their homework online usually, so they can't see what their answers were. They're just trying to remember what it was they put. All right. Do you remember for this virus, what's this top thing? Yes, it's a head. And what's in the head? DNA is in the head. Very good. And then what's this little thing called in the middle? Do you remember? Collar. Good, Everly. And I was explaining that if they answer questions correctly, they get a little coin in the cubby and they use them on the way out the door to get candy. It's pretty cool. All right. What's this big long part called? Does anybody remember that? Okay, that's going to be your sheath. And what's this little guy at the bottom? Do you remember that? Very good, base plate. And then what are these guys called? Tail fibers. And I think this is a UK resource because it has fibers spelled not the way we spell it here. All right, so let's move on to the lesson. So what is a protist? Today we're talking about protists, protozoa, protists. And we'll be looking at 3 different kinds of protists in lab today. And they all have a different means of locomotion. So it's really, and a different means of feeding too. So it's really kind of fun to see how different they are. So do you remember what a eukaryote is? It's a big one. You should know this one. A eukaryote is any cell that has a what? Nucleus bound by a membrane bound nucleus, right? If you don't have a membrane bound nucleus, you're not a eukaryote, you're a, do you remember? Prokaryote. Good job, gang. All right. So any single-celled or multicellular organism whose cells contain a distinct membrane-bound nucleus is a eukaryote. Are you a eukaryote? Yes, you are. A protist is any unicellular eukaryotic organism that is not a plant, animal, or a fungus. All right. Unicellular means what? One cell. If you didn't know that, how might you figure it out by looking at the word? What's uni mean? One cell. Very good. Remember, we do that a lot. When we see things that have Greek or Latin roots in them, we pay attention to them because sometimes they can tell us what the word means. Okay, so if it's not a plant, animal, or fungus, and it's unicellular and eukaryotic, it's going to be a protist. But some protists are multicellular. Okay, so it's kind of like a Venn diagram situation. So all protists live in moist spaces. Most are unicellular, but some are multicellular. Some can move and some cannot. While it's likely that protists share a common ancestor, the exclusion of other eukaryotes, meaning the plants, animals, and fungus, because they are not grouped with them, means that they don't form like a natural group or clade with them. So just evolutionarily, how we're speaking is these are kind of an offshoot. They don't necessarily follow along to other things. And they can be very, very diverse, meaning lots of different types. So some are more closely related to plants, animals, or fungi than they are other protists. So we'll see in a minute how that looks. So here are some that are more similar to plants, and then we're going to talk about some that are more similar to animals and some that are more similar to fungi. So green algae are the closest relatives to land plants. All right. So if you talk about land plants, what do we mean? Like all these guys that we see outside every day, right? So these are they're related closely. So evidence shows they evolved into land. So these went into land plants about 475 million years ago.
Somebody they started in seedling?
Yeah, kind of. So the way like an evolutionary tree works is sometimes you'll have organisms that will branch off and then they'll keep branching this way, and then others that'll branch later or earlier. So it doesn't necessarily mean that every plant that you're going to find came from a particularly straight line. So it's kind of hard if you have a visual, which we've had before when we've had this discussion. I just don't have one at the moment. But yes, in general, to your question, as a generalization, yes, but there were going to be some exceptions. So a long time ago, 475 million years ago, right? So green algae are split into 2 main groups. We have chlorophytes and charophytes. And both groups are single-celled, and they contain single-celled and multicellular algae. So you know what algae is, right? We've done, what lab did we do with algae? Do the 3 of you remember? It was way back at the beginning of the year when we were doing ecology stuff. Remember, we took the little single-cell green algae and then we added something to it. Do you remember what we added to the water? Remember, we added fertilizer to see whether or not it grew more, right? because that is an issue. Farmers will fertilize their land, that water goes into runoff ponds, and then you have so much algae that it can like cover the surface of the water and it makes it harder for some of the organisms underneath to get the light they need, or sometimes even the oxygen they need, because they can choke it out.
Sometimes
Absolutely, yes. And we'll start a little pile for you since you've contributed. All right, now charophytes are the most closely related organisms to land plants and are found in freshwater environments. And then chlorophytes are found in marine freshwater. So this is kind of what you were addressing. And land-based ecosystems are most commonly found in shallow freshwater. All right, now let's talk about those that are more similar to animals. So protozoa is an informal term for a group of single-cell eukaryotes, either free living or parasitic. What would the difference be there? Free living or parasitic?
there's something that lacks on something else.
Yeah, it needs something else in order to live. It needs another living organism in order to live. And in this case, in particular, in that relationship, the parasite benefits to the detriment of the other organism, meaning the other organism does not benefit. It's actually harmed usually. There are different types. There's commensalism and mutualism and different types of I don't want to say different relationships between organisms, but in parasitism in particular, one's going to benefit to the detriment of the other. And they can feed on organic tissue, debris, things like that. Historically, protozoans were regarded as one-celled animals because they have animal-like behavior, but now we've categorized them differently. And some of those behaviors are things like the way they move. Can they be predators? And they lack a cell wall, which is found in a lot of found in plants and also found in algae. So these don't often have a cell wall. And then historically, they're divided into four major groups. The amoeba, this is spelled differently here. You're going to see this word spelled differently. Sometimes there's an O in that word. So it's a weird O. It's like a silent O. It doesn't make much sense. We don't see those often. But anyway, the amoeba, the flagellates, the ciliates, and the sporozoa. And today in lab, you're going to be looking at amoebas. And I will give you a heads up. I'll tell everybody again before lab. They are very elusive. They are hard to find under the microscope slide. And they move very slowly. And sometimes it's even hard to see them moving. The flagellates, which are the guys with the little whippy tail. We talked about these last week, right? Some bacteria have them too, like the whippy tail, and they move around real fast. We'll be looking at Euglena. They have a flagella. So they move real fast. Hard to miss. They move so fast. And then ciliates, we'll be looking at paramecium. They have almost like little hairs on the outside of their body that they beat. It's kind of like if you ever seen a crew team, a row team, where you've got like a bunch of guys in a boat and they all row together. You ever seen that? Yeah, it's kind of like that. And then also sporozoa, which are a little different in how they work. We won't have any of those to look at today. Then we have fungus-like protists we call molds, and these are absorptive feeders. So they have to have decaying organic matter in order to eat. And they resemble fungi and they reproduce with spores as fungi do. Okay, so look, these guys feed on decaying matter like fungus, right? How do these guys get food? It's right there in the name. What do you think? What does that word remind you of? Chlorophyte. Does it remind you of a word you've heard before? Chlorine. Maybe. What what's the green stuff inside of plant cells?
Or
Chlorophyll, right. So those guys actually do produce. These would be autotrophs, meaning they produce their own food, right, from like solar energy in that case. And then these guys are more like your predatory types. They eat, quotes, air quotes, eat other things, right? All right. So in other ways, these particular fungus-like protists are quite different from fungi and more like other protists, so they can have a cell wall which is made of cellulose instead of chitin. So they will have cell walls, but it's made of something different. If you remember, plant cells are made of cellulose, and casually in our day-to-day speak, we call that fiber. Like, remember we've talked about that before when you need a high fiber diet or you need to eat extra fiber, that's all you're eating is plant cell walls. It's cellulose, right? But chitin is what the cell walls in a fungus is made of. It's made of chitin. Also, interestingly, chitin makes up arthropod exoskeletons. So like ants, grasshoppers, things like that is chitin again. So it's the same material. And in like other protists, they have complicated life cycles. They can reproduce sexually or asexually, and they are motile sometimes. They can move during some stages of their life cycle. And we have two major types. We have slime molds and water molds, just what they're called. So we're going to watch some video of them moving down here as we talk about them. So these are the three you'll be looking at in lab today. A paramecium and amoeba. See, there's that O. I told you it shows up sometimes. Two correct spellings. You can go with either one. I usually do the O. Yeah, English is real fun, right? And then this little flagellum that flips around while they swim. So most protists are motile, but different types of protists have evolved varying modes of movement. So protists such as a euglena have one or more flagella that they rotate or whip. So you see how quickly they move? That's what they're going to look like under your microscope today. You're going to see them moving real fast. You're actually going to take some little itty bitty like fibers of cotton and put it under your cover slip when you make your slide because it kind of pins them in. If you can make kind of pins them into a place and slows them down. Otherwise, you can't really observe them. They move so fast. Then you've also got paramecia, which would be your plural of paramecium, and they're covered with rows of tiny cilia that beat to swim through liquids. So you see how they move? Look. They remind me of remote control cars because they stop and back up and move like really quickly. It's very abrupt, their movement. And then other protists, such as amoebae, which would be your plural of amoeba. Some people are just going to say amoebas. I'm probably going to say it because that one has sound. Anyway, they move using pseudopods or pseudopodia. So if you look real close, do you see you see those little dots inside that are moving? That's about all the movement you're going to see today in an amoeba if you manage to catch one. I have one that I did manage to nail down this morning finally after days of fun. I looked at it several times over the last few days and didn't get lucky. And this morning I did. Finally found one.
Like ball things.
So that's an amoeba. And whoop, that's another protist that doesn't belong, probably paramecium. But that amoeba, what it does is its cell wall is real flexible and it will move its cytoplasm and it will like stretch and crawl and sort of move along as it goes. It will also use its pseudopod means false foot. So that's what it does. It creates a foot and it kind of pulls itself along. But it will also use those pseudopods if it finds something it wants to eat and it'll wrap them around it like this and like a hug, like a hungry hug and pull it in. And then lastly, hi, come on in. I think we got somebody make a little space. Make yourself comfortable. Now, what's your name? Grant. Okay, Grant. All right. Well, we're a little bit in. We're talking about protists today, which are a particular type of usually unicellular, but not always life form. We're moving from our most basic to our most complex life forms in this unit. All right. So then lastly, we want to talk about taxis and phototaxis. All right, so some are going to move away or towards certain stimuli. Now, I don't know if any of the three of you that are in this class remember, but phototaxis was a word that we used when we talked about you remember when we did the flatworms, the planaria, the little worms? And remember we cut them and they like regenerated body parts. But do you remember how they behaved in reference to light? Do you remember? So if you put a bright light on them, they move away from it. Okay. And that is a type of phototaxis. Taxis meaning response to a stimuli, moving toward or away. And photo means that it's light that causes them to behave that way.
Why is it light?
Why? They do better in darker environments. That's where their food is. And they also it's just kind of they thrive out of the light. The light isn't necessarily a healthy situation for them, tends to be warmer or whatever. And they have little eye spots. They have two little, looks like eyes, but they don't really see. The eye spots are just to tell them whether they're in the light or the dark. So let's talk about some plant-like protists, and I have a video for you too. So protists that perform photosynthesis or autotrophic, what's autotrophic mean? For those of you who are in this class, 3 of you are actually in this class. Come on, heterotroph, autotroph, chemoheterotroph, we've talked about this. What's autotrophic? Autotrophic means it produces its own food from a source like the sun, right? Doesn't need chemicals. Heterotroph gets its food by eating other organic matter, eating other organisms, right? Or dead organisms. All right, so they're autotrophic because it's photosynthesis, so it makes its own food. The position of photosynthetic protists at the base of the food chain makes much of the diversity of aquatic life possible. They feed fish and whales, support coral reefs, and provide shelter to marine life. In areas where sewage is dumped, protists help recycle waste. However, when the amount of waste is excessive, algae grow into enormous masses called algal blooms, which we talked about.
Thanks to the Monterey Bay Aquarium for partnering with
Now, I told y'all before that the rainforests are not the lungs of the planet. It's actually the ocean. And this is why. Hank will tell you, little Hank.
Right now, there are incredible creatures in the ocean with the power to reshape the planet's atmosphere. After all, they've done it before, and you've probably never even seen them unless you've looked at seawater under a microscope. I'm talking, of course, about phytoplankton, the microscopic critters that almost all life in the oceans and arguably on land depends on. Phytoplankton is a catch-all category for all the tiny aquatic organisms that can turn sunlight into food via photosynthesis. And there are all kinds of different organisms included in it, from cyanobacteria to whip-tailed dinoflagellates to armored diatoms, which look amazing under a microscope, by the way. Diatoms are essentially algae that build little houses out of glass, or silica to be precise, and they come in a stunning diversity of forms. In fact, if you've ever spread diatomaceous earth around your garden or your house, you were sprinkling fossilized diatoms. There are also coccolithophores, which can look kind of like a soccer ball covered in frisbees thanks to their teeny tiny shells made of calcium carbonate. In fact, England's famous White Cliffs of Dover contain the fossilized remains of countless coccolithophores. And in today's ocean, the smelly compounds they produce help give the sea its unmistakable scent, a smell birds like albatrosses use to find their meals. The compounds that make those smells can also travel up high into the atmosphere to help clouds form and grow, making them more reflective and helping to keep us down here a lot cooler. And dinoflagellates are also very cool, don't want to leave them out. They have two tail-like flagella that help them swim around, and many can produce their own light, which makes for some really magical effects. But too many of them in one place can create a harmful algal bloom, a phenomenon you might have heard referred to as red tide, which is just one example of how these super teeny creatures can have a big impact on the rest of us, even us landlubbers. No matter what form phytoplankton take, they are unbelievably important to all life on Earth. First off, they're the foundation of the marine food web, underpinning everything from anchovies to whales. In fact, with a wee bit of help from seaweed and aquatic plants, they managed to support life in the ocean despite being outweighed by roughly 5-to-1 by organisms that don't make their own food. And if that's not enough, they are also atmosphere-altering machines. They consume about as much carbon dioxide as all of the world's forests and other plants combined, pulling roughly 11 gigatons of carbon from our atmosphere every year, which is especially impressive when you consider that by mass, there's about 450 times as much plant life on land. Plus, phytoplankton account for more than half of the planet's yearly oxygen production. But even more importantly, they're kind of the reason we have oxygen in the atmosphere at all. Back about 2.5 billion years ago, near the boundary between the Archean and the Proterozoic eons, the Earth was a very different place. There were green seas and orange skies, and the atmosphere was likely a mix of gases like ammonia and methane that would immediately kill us. Then cyanobacteria evolved and ran rampant, pumping out a huge amount of oxygen in what's now called the Great Oxidation Event. All that oxygen helped turn the ocean and skies blue and likely jump-started the evolution of complex life. And then around 540 million years ago, phytoplankton did it again, exploding in numbers and bringing us up to more or less the levels of oxygen in the atmosphere we have today. Paleontologists think the boom may have occurred thanks to nutrients weathered out of rocks by giant continent-sized glaciers. A recent idea is that it happened because the plankton evolved a more efficient internal machinery related to photosynthesis. Either way, this set the stage for life to fully spread throughout the ocean and onto the land. Since then, they've calmed down on the reshaping the entire planet front and adopted more of a stabilizing role. For example, paleontologists think the evolution of calcifying plankton balanced ocean chemistry and helped protect the seas from mass extinction. Before about 80 million years ago, the ocean was not a super stable place to be. Changes in the chemistry of the atmosphere from events like volcanic eruptions often caused deadly swings in seawater pH. Then some lineages of plankton, including those ball-like coccolithophores, figured out how to make shells of calcium carbonate. Soon, they took over the open ocean. And over a relatively short period of time, so many of their shells sank to the depths of the ocean that they created a vast, thick layer of calcium carbonate ooze on the seafloor. That ooze acts as a chemical buffer in the ocean, a natural braking system on runaway acidity. See, the shells are solid at a more neutral pH, but as the water gets more acidic, they start to dissolve, releasing carbonate ions. Those chemically react with the surrounding seawater to push an acidic pH back towards neutral. Now, there had been a minor version of this in the shallows, thanks to mollusks and other large-shelled organisms, but it took phytoplankton to make it ocean-wide. And the end result was a more stable ocean chemistry, which meant marine life became less vulnerable to things like volcanic eruptions. Even today, this chalk layer is helping buffer the acidifying effects of all the carbon dioxide that we humans are pumping into the atmosphere. Though there's only so much it can do. When we consider everything that lives in the ocean, from majestic whales to bountiful coral reefs, these little unassuming phytoplankton are easy to overlook. But if you look at the history of Earth, it doesn't take long to realize that though they are small, they are also mighty, and they deserve as much fascination, love, and protection as any other living thing on this planet. Thank you again to the Monterey Bay Aquarium.
Yes, Monterey Bay. So you mentioned diatoms. I'm wearing diatoms today. I love them. They're so pretty, so cool. Yeah, so we talk about this a fair bit in earth science. One of the things that we do in earth science is we compare and contrast some documentaries that are you switching places? Trying to we can, what is it?
A ladybug.
Oh, it's not a real ladybug.
What?
Nope. Nope. These have, where is it?
What?
Where is it?
It's on the.
Oh, it's underneath. I'm gonna borrow it. Because if it were a real ladybug, I would put it outside. But these are, may I grab one? Thank you. These are imposters, and they are from Asia, and they are not indigenous, and they are not part of our ecosystem. So here's an example of what we do with them. If it was one of our native ladybugs, I would take it outside and free it, but it is not. So it doesn't. You can, if you Google them, side by, they're called Asian. Oh, hang on, I can't remember what they're called exactly. If you look up imposter Asian ladybug on Google, you'll find it. And you can see ladybugs side by side. I've got a couple of them up there. They've made their way in and I vacuumed them up, and the next day more of them are here. And I don't know why they're here, but anyway, they are like the Joro spiders and the brown marmorated stink bugs that do not belong here, are not part of our ecosystem, really shouldn't be. So we talk in earth science about, we compare and contrast some climate change documentaries for the purpose of understanding how to take that data and information in, and then how to analyze it and how to form your own opinion on what you're seeing. And we watch 2 in the spring, 2 in the fall, and 2 in the spring, and they oppose each other. The first one is going to give you one message using data, and the second one is going to use the same data and give you a completely different message as to what's happening. And you have to try and figure out what you're seeing, how you determine one from the other, and we walk through it. And I never tell you what your opinion should be, but it's interesting to compare and contrast them. And that is one of the things that comes up is deforestation. And we've talked about this before. Deforestation isn't good for a number of reasons. But like every so often, there'll be a fire outbreak in the Amazon, which is terrible for a number of reasons. But you will see a lot of media reports that the lungs of the earth are dying. And if we don't have the Amazon rainforest, this little patch of Amazon, oh, we're all going to suffer. There's a lot of reasons why it's bad, but that's not what is providing most of Earth's oxygen. It is coming from phytoplankton in the ocean. There's some great videos where you can actually, they sort of map the way it moves like out of the ocean, and there's like some currents that are almost like rivers in the air and how it moves. So, I mean, definitely we don't want deforestation and we don't want uncontrolled forest fires for various reasons, but it's not the apocalyptic end of the world due to lack of oxygen, as some people would have you believe. It's due to other things that it might be bad. All right, so let's talk about these slime molds. So these obtain energy by decomposing organic material, and as a result are important for recycling nutrients. They can be brightly colored, live in cool, moist, dark habitats. They're classified as either plasmodial or cellular based on how they reproduce. Now, the plasmodial ones, they have no cell wall situation going on. It's just like a blob of cytoplasm, little dotted with little nuclei everywhere. It's kind of interesting. Whereas the cellular ones are, sort of divided into more individual cells, the way you think of traditional cells with cell membranes. And they can engulf food like amoebas. Now, it's another video. I usually don't do videos back to back, but this video is way cool. So this is a slime mold, and you're going to watch this slime mold solve a maze. And not only does it solve a maze, it does something else with mapping at the end of this that's actually really, really fascinating. Now, remember, this doesn't have a brain, right? It can't think, it can't reason, it can't make decisions, but it can still solve a maze. Oh, oh, cute dog. Okay. I like the dog. Skip. Okay, Ad.
At Oxford University in England, Dr. Mark Fricker is one of a team of botanists and computer scientists studying a form of slime mold called Fusarium polycephalum. For years, slime molds have fascinated scientists with their ability to navigate the quickest route through a maze to get at some food. But scientists started to wonder if the slime mold could do more than just perform clever tricks. Here, Mark is recreating an experiment he worked on with colleagues at Tokyo University. A blob of slime mold is surrounded with a pattern of oat flakes, an irresistible treat for the slime mold. What happens next is recorded by a time-lapse camera.
So the slime mold starts as this blob in the middle, and then it's going to spread out and colonize the whole of the dish, trying to find all of those oat flakes that we positioned earlier on. So connect each of those food resources, but some of those links will disappear, and some of them will be strengthened.
What the slime mold has done is truly remarkable.
The way we set out those oat flakes was not entirely random. The central blob is supposed to represent Tokyo, and if we positioned all of the oat flakes on the major cities around Tokyo, so what we're now looking at is not just the slime mold exploring a random distribution of oat flakes, it's actually trying to map out the region around Tokyo to see how the slime mold would connect all of those cities to compare against the way humans have connected it with their own network.
When you compare the local Tokyo rail system and the network that the slime mold has come up with, the two designs
That's pretty
are
amazing, right?
remarkably similar.
It did what we would do.
In just a few hours, the slime has done what skilled engineers took years to achieve. It has found the most efficient way to link together multiple locations, every bit as effective as the well-organized Tokyo rail system.
The slime mold doesn't have a brain or any way of calculating this, but has managed to produce a network with very similar properties.
That's pretty wild, right? It did exactly the way we would do it. It found the most efficient routes, which is what we've done. So that's pretty crazy. We're still learning how it does that. All right, so let's go through here with some quick answers. Let's see what we can do. So protists reproduce by a variety of mechanisms. All right, with asexual reproduction, most undergo some form of asexual reproduction, such as what? Which produces two daughter cells. What's it called when you get two daughter cells? Anybody have their binder open for that answer? That's going to be binary fission, binary meaning two, fission meaning dividing, dividing into two. Then we've also got some protists, such as true slime molds, exhibit what? Which simultaneously divide many into many daughter cells. Do we know what that one's called when you get many daughter cells? Looking. That one's multiple fission, so dividing multiple times. Others undergo this process where you get tiny little buds that grow and divide grow to the size of the parental protist. The word's in there, it's a clue, that's budding, right? And then fungus-like protists were once classified as fungi because they produce sporangia, and that one's right there, a sporangium is a structure that produces and contains spores for asexual reproduction. Now, as far as those that reproduce sexually, there's the answer. Sexual reproduction involves meiosis and fertilization, is common among protists, and many protist species can switch between sexual and asexual reproduction. Why is that advantageous? Why might you want one over the other? Say you're alone and you don't have any other organisms to reproduce with, then you're going to want to reproduce asexually, right? But if you want to vary the gene pool, that's where sexual reproduction is useful. And what's another type that mainly occurs only in ciliates? This one is called conjugation. And ciliates, such as paramecium, they will like bump up to each other, and there will be like a dissolution of the cell membrane, and they will exchange DNA or genetic material and then divide and move away again, which is pretty cool. All right, so let's talk about fungi for a bit. So fungi are eukaryotic heterotrophs. Heterotroph, meaning they need another source for their energy. They do not produce their own food. Cell walls contain chitin, we said this earlier, and which is a complex carbohydrate. They cannot make their own food. They have a nucleus and a cell wall composed of chitin. They grow in warm, moist places. They can be unicellular or multicellular. Most fungi are composed of thin filaments called hyphae. Now, when you think of a mushroom, right, you think the mushroom is the fungus, right? It's really not. Most of the fungus is under the ground and you don't see it. It's part of the mycelium or the hyphae. What you see above ground is called the fruiting body. It's just the reproductive structure of that particular fungus. So that mushroom is just a reproductive structure. That's all that is. All right, so the fruiting body of the fungus, such as the above ground part of a mushroom, is a reproductive structure that you can see. It grows from many hyphae tangled underground in a thick mass called a mycelium. Hyphae ooze chemicals that break down and absorb food in the environment, and they absorb nutrients. Most fungi reproduce both asexually and sexually. Asexual reproduction can occur when cells or hyphae break off and begin to grow on their own. And some fungi also reproduce asexually by means of spores. And spores, a spore that is produced by a mushroom, it's in the gill part of the mushroom, which is what you're seeing here. That part, this is the underside of a mushroom, right? And we're actually going to do something with them in a minute that can be kind of fun. And anyway, those they'll produce little spores which will fall on the ground or blow in the wind and land somewhere else, and then you'll get a new fungus in that area. So, does anybody know what that is? It's a fairy ring. You ever seen one? I love these. They're a natural phenomenon. Nobody planted this. This is what happens. Sometimes it's not a complete circle, but it's a natural circle or an arc of mushrooms, and they've got often greener or dead grass. The grass around it can be affected by them. And it's formed by an underground fungal growth, or the mycelium, which is what's underground. It expands outward from the center, which is how it makes a ring, right? Expands outward from the center, and all the little fruiting bodies pop up around the outside. And the ring's edge, the fruiting bodies emerge at the ring's edge as the fungus feeds on decaying organic matter in the soil, leading to either lush growth of grass or nutrient depletion in grass, depending on what that grass needs. But if you look, especially in like when the spring and in the fall, when it's not too hot, but it's warm in the morning, if you've had some rain lately and the ground's nice and wet, you might see these pop up in places. I have a neighbor that I pass. I don't know them, but I pass their house all the time. And they have this, like nearly every spring, there's one that keeps popping up in their yard. And I know they're trying to kill it and it keeps coming back. But it's beautiful. Theirs isn't a full circle, it's an arc. But it's called a fairy ring. Does anybody know why it's called a fairy ring? You want to guess? Is what? It's a circle. That's the ring part. So the idea, the romantic idea, is that little fairies would come out at night and they'd sit on the little toadstools and they'd have a little meeting because it's a nice little circle. That's a great idea, right? I would love that to be the case, but it's really cool. All right, now here's the cool thing about mycelium. You don't see the mycelium of the fungus, it is underground. But not only is it essential to the fungus itself, it helps the trees in the forest communicate with one another. It's like the telecommunication service of all the trees. And it's really, really cool. And this is something that we're still just kind of learning about. It's kind of new. Oh, another commercial. Work out, be strong. Planet Fitness. Can never get rid of these things.
Best PB&J.
Now I want a PB&J.
Made simply.
There we go.
The idea of talking trees has been capturing the human imagination for generations.
My back is worse than my butt.
Okay, so maybe they don't talk to us, but it turns out that trees can talk to each other.
Trees are speaking to each other.
That does beg the question, what do trees have to talk about? And can we learn to speak their language? Underneath the soil, a vast and interconnected network of life links the trees through their root systems. But they can't talk to each other without help. The whole process starts with hub trees, the oldest and tallest trees in the forest. Hub trees have greater access to sunlight, and through the process of photosynthesis, end up producing more sugar than they actually need. Underground, fungi need sugar to survive. Most of their bodies are made up of a mass of threads called mycelium. They grow within the root system of trees to absorb the excess sugar. In return, the mycelium provides the tree with the nutrients it needs from the soil. This symbiotic relationship is known as mycorrhiza, which stems from the Greek words for fungus and root. These tree-fungi relationships connect the trees in the forest together, forming an underground communication network to exchange water and nutrients, nurture their seedlings, and even send warning signals when under threat. So, how many trees are really talking to each other? To get a better picture of these forest relationships, a team of researchers used DNA analysis to map a fungal network in a patch of Canadian forest. Remarkably, they found that one tree was connected to 47 other trees. Their models also showed that when hub trees were removed, it would cause more connections to be lost than if trees were simply removed randomly. Studying these kinds of underground exchanges will play a vital role in creating stronger, more resilient forests for the future. So even though we might not be able to talk to trees, at least we can still keep trying to understand their language. Who knows what they might say?
So that's interesting. If you remove the hub tree, it's going to have a detrimental effect on how everybody else is communicating because they're all communicating through that tree. Did you know the trees talk to each other like that? Kind of cool, right? Really interesting. That's right. So we're going to real quick look at the diagram of a mushroom, and then we have a quick activity that we're going to do before lab. So here are your parts, your scales, your cap, your gills, which are going to be concerned with in our little activity of the gills, rig skirts, stipes or stems, volvas, and mycelial threads. If you've ever popped a mushroom out of the ground, you've probably seen these guys, right? We're gonna skip this one, but you can watch it on your own. It talks about how some mushrooms glow, and they do. Some mushrooms are bioluminescent, and they're trying to figure out why. So that'll be in your weekly email and in your resources. Okay, so here's our activity. What we're gonna do is we don't do little crafts too often, but this is a little craft. So I'm gonna send you to the back of the breakfast room in a minute. The moms are gonna get bombarded, but what you're gonna do, I'm gonna tell you what you're gonna do first. I have to go grab them out of the fridge, but I have a bunch of mushrooms that I've popped the stems out of, and I've carefully trimmed the bottom so that the gills are exposed. And what you're gonna do is you're gonna take a piece of paper, write your name on it, flip the paper over to the blank side. And then you're gonna pick, since there's extras of you today, I'm going to say 1 to 2 mushroom caps instead of 1 to 3 because I don't want to run out. So 1 to 2 mushroom caps, and you're going to place them. Those of you that aren't staying, I'll give you bags to take them home. You can do this at home because you won't be back, right? Or at least not tomorrow, next week. Hopefully you will be back, but not next week. And you're going to place them gills down on the paper, and I'm going to leave them there for a couple days. And then what happens is the spores that are in there will fall out, and they will make a print. And with any luck, it'll look similar. It's probably going to be much lighter in color, but it'll be there like that. And then you're going to take it home. When you come next week, you're going to take it home, decorate it, turn it into something, and bring it back, and I will display them for everybody. Cool? All right, so y'all want to hop up and move that way. I'm going to grab those mushrooms for you. Sorry, Moms, I know you were going to stop pulling out and doing it. You can grab it. Sorry. All right, so y'all put it right here. All right, so name, pick 1 or 2. Those of you that aren't coming back next week that are visiting, I'll give you a Ziploc bag to take your film in, but you can take a piece of paper and put your name on it afterwards. It's just everything I'm used to. Get grab this. Give me a one. You can pop yours and take this paper. You can home. There you go. And then you can just leave them on that table. Evan, Everly, and Emerson, you can leave them on that table there. And then I will I'll deal with them later then. I don't wait, are we gonna get some paper? Or take them home because they need to rest for a long time. If you put it leave paper. Yeah. Right. You take all of this and then and then when you get yeah, when you get home, you just turn them over and leave them for a day or 2, and then pick them up, and you should have a print. That's what I'm gonna do. And all the rest, y'all can move in this way, giving them a second to finish up an activity, and then we'll start. Okay.
I need to put a chair there.